Giometto. He's in civil environmental engineering, and his background is kind of diverse. He has undergraduate degrees in physics, his bachelor's degree, and his master's degree is in theoretical physics from University of Padua. And then he went on to get a PhD in civil and environmental engineering at the École Polytechnique, the Swiss Federal Institute in Lausanne. Okay, and then he went on for a postdoc at Harvard in physics and also in cellular and molecular biology. And then in that wonderful quiescent, highly normalized time that was summer of 2020, he elected to join my department and came and is with us now. I highly recommend that you go visit him at his office and talk to him for two reasons. One, he's really interesting and has a great background for technical discussions. And two, he has a fantastic espresso machine. He's such a good host that he'll make you a really, really nice cup of espresso, I promise. So with that, I'll turn it over to Andrea.
Thank you so much. Yes, everyone is welcome. I have 4 cups, so more than you bring one.
Thank you.
Perfect. So, well, thank you so much. I'm so happy giving an in-person talk finally after many, many months. So, as you can probably tell from the title, my interests are in the evolutionary dynamics of populations and ecological dynamics as well. And often when we talk about evolution, this is the first thing that people think about. This is probably because these organisms look much different than the ones we see around us today. It's probably because traditionally, until a few decades ago, if we wanted to study evolution, we had to look at the fossil record or comparative studies. And of course, partly it's because of Jurassic Park. Nowadays, this is where I think we can really understand evolution much more. And I'm not the first one thinking about this. In fact, it's been a few decades that evolutionary biologists are using microbial microcosms to understand evolutionary dynamics. Now this here is E. coli, it's a unicellular prokaryote, and Richard Lansky and his lab have been propagating cultures of E. coli that look like this. So every flask here, you can think about it as a separate universe, that all these flasks started with the same cell. And what they are doing there is, since 1988, every day they've been diluting this population into fresh medium. So they have been propagating every day this population. These cells do 6 generations every day. And so at this point, they have surpassed the 70,000 generation. Now, 6 generations in human times is approximately 150 years. So you can imagine this has been going on for a very, very long time. And looking at these systems can give us a view into evolution. And we can use this system to understand evolution as a stochastic dynamical process, in which we have mutations arising in these populations. And these mutations can either establish and reach the majority of the population. In population genetics, we say the mutation fixes in the population. Or they might go extinct. Different mutations might be beneficial or deleterious, or they could be neutral, not giving any advantage. Now, these populations have been changing. They are different now than they were in 1988. For example, if you look at the growth rate relative to the ancestor, meaning the ancestor cell at time 0 had a growth rate of 1. Now, the growth rate at the 70,000 generation, which is around here, is about 1.8 times. So if these cells took 1 hour to divide back in 1988, it now takes them only 40 minutes because they became much more adapted to their environment. So this is, oh, and I should say, please ask questions during the talk at any time. Interrupt me whenever you want. So, okay, this is what we say, it's a phenotypic. It's a phenotype, right? They are growing faster, but underneath this phenotype, there are many, many different mutations that have occurred. And a few of them have fixed and have caused this phenotype, have caused cells to become faster. Now that we can sequence things easily, we can look for every mutation that reaches a detectable frequency. We can plot their frequency in the population. So some of them, like this one, rapidly swept through the population and fixed. it means that now every cell in the population has this mutation. Other ones arose, increased their frequency, but eventually went extinct. And from here, you can also see that the right part of the graph is way denser than the left part of the graph. And what happened here is that at some point here, there was a mutation that increased the mutation rate. So in a sense, that mutation changed the evolutionary dynamics. They're now exploring the space of possible genotypes much faster than they were before. So this is all great, especially with these new sequencing technologies. We can really get a lot of detail about the evolutionary dynamics in these simple communities. But on the other hand, these communities are maybe a bit too simple. They are just flasks with just one strain of cells that's agitated all the time. So you can think of these cells as they are all interacting with all the others. Very often, microbial populations are either dense or spatially structured. You can think of biofilms, you can think of microbial mats such as the ones that you can see at Yellowstone. You can think of human infections and many, many other scenarios like engineering applications. We're starting to understand that the fact that populations have spatial structure, or the fact that populations are associated with surfaces, or the fact that they are very dense, has a strong impact on their dynamics. These populations look quite different than populations that are well mixed, as if you may think the ocean might look like. So I'm interested in understanding how spatial structure and how the density of microbial strains affect their evolutionary dynamics. And so I will show you today two main projects that I worked on. The first one concerns how mechanical interactions among dense populations of cells reduce the power of natural selection. I will say what I mean by this. I will briefly talk about some work that we're doing in the lab right now, where we're looking how populations evolve to expand faster on surfaces. And I'll show you that cell size has something to do, cell shape has something to do with it. And finally, I will talk about how biological interactions among strains of microbes can affect their evolutionary dynamics. And in particular, I will show you how cells that are engaged in warfare are an analog of nucleation dynamics on a petri dish. So, part one. Now, this here is the budding yeast, Saccharomyces cerevisiae. We like to work with the budding yeast for mainly because yeast geneticists for the last few decades have been discovering tools to modify the genome of these cells. And so we can play around with it in the lab, and we can make these cells do almost whatever we want. Now, these cells are non-motile, so they don't have cilia, they don't have flagella. If you put them someplace, they sit there, at the very most they will do some kind of Brownian motion. These cells divide by forming a bud on the cell surface that grows as the cell progresses through the cell cycle, and eventually the bud will separate from the mother. And now the fates of these two cells are somewhat independent. What the easiest thing probably we can do from the genetic point of view is we can make these cells express different fluorescent proteins, so that if we look at them at the fluorescent microscope, we can distinguish blue cells, for example, from yellow cells. These two cells are completely identical. The only thing that's different is this one produces a blue protein, this one produces a yellow protein. And why is this interesting? It's interesting because by coloring cells, we can follow the fate of cells in space and time. We can follow lineages. So here's the simplest experiment we can do. We can take these two cell types, we can mix them up in a liquid culture, we can take a droplet and we can deposit it on a surface of an agar plate that contains nuclei. This is how the initial condition looks like. You can barely tell that there's a population of cells here. And if we follow it with time, this is what we see. We had a population initially that was concentrated here in what we call the homeland. that population was well mixed because we had agitated the tube before. But you see, as the cells divide, they pile up and they expand, they form a colony that expands radially. As the colony expands radially, the two colors separate in space. This is an analog of a loss of what we call genetic diversity. You can think of these colors as an allele. It could be people with brown hair or people with long hair. As it expands, the two colors separate, just due to the fact that the mother cell will give rise to a daughter cell nearby. If we zoom in, now I'm going to show you a zoom in of the initial condition here. If we zoom in, this is how it looks like. Now, every circle here, every disk is an individual cell, and these, you see we have both blue and yellow cells. If we record what happens in the first few phases of the dynamics, what you can see is that cells are dividing. As they divide, they push each other. This is a very, very dense colony. As they push each other, some cells will fail to form a sector. For example, if you look at, if you look at this blue cell, it will fail to establish a sector up here just because right now, due to chance event, it's being closed off. The yellow cells have prevented it from reaching the front. And so this is the kind of coarsening dynamics that eventually produces the separation of colors. Now this is cool to watch in the lab, but it has some implications for how we understand the world around us. For example, it is thought that the same effect is responsible in humans for the loss of genetic diversity as we go from Africa to Northern Europe. It is thought that as the humans expanded out of Africa, we had a similar situations in which we had many different alleles, many different colors that got lost as the population expanded, just like the yeast cells segregated into areas of different colors. So this is all good, but it's a very simple experiment. We would like to know something more. We wanted to know, for example, how fast a fitter genotype would expand in the population. By which I mean, suppose we do an experiment with a cell, a strain that divides fast and a strain that divides more slowly. We would like to know how does the faster growing cell expand its relative abundance as time goes by. And conversely, we would like to know how do less fit genotypes go extinct. If we have a well-mixed population, for example, yeast cells in a culture tube that is well-mixed, we can resort to classical population genetics to answer this question, and we have a relatively good idea of the forces that control which strain we establish. If we are dealing with a spatially structured and dense population living on a surface, it turns out we didn't really know much about this problem. And so now I'll show you an example in which we try to answer partly this question. We repeated the exact same experiment I've shown you a video of, but this time with two different strains, a blue one that grows slowly, so we say it has low fitness, and the yellow one that divides fast, it grows fast, so we can think that in the same amount of time, the blue cell will give rise to 2 daughters, and the yellow cell will give rise to 3 daughters, for example. So we expect yellow cells to increase their relative abundance, their frequency in the population. So we start the experiment with only 1% of yellow cells. This is how the colony looks like after 1 day. It started out as a droplet somewhere here. You see, after 1 day already, we start having the separation of genotypes, separation of colors at the edge. As we let time go by, what happens is some of the yellow sectors fail to establish. Even though it's dividing faster, stochasticity is still important. Some of these sectors will not make it and will be closed off by blue cells. But some of them do establish, and they form these yellow sectors that bulge out from the colony. So the first thing we wanted to understand was, okay, what is the shape of this sector? Like, can we characterize the shape of this sector? The reason why we would want to do that is because it tells us how fast the yellow cells, the fitter cells, increase their frequency in this population. So we were not the first one to think about this. It turns out that Kirill Korolev and others had thought about this from a very abstract point of view. So the way they thought about this is inspired by geometrical optics. So they thought, let's think about this colony where we have blue cells here and faster dividing yellow cells here. And let's assume that it's only the cells at the very edge that are actively dividing. So we can think of this system as follows. We can assume that in an infinitesimal time step, blue points will give rise to circular expanding waves with speed vd, and yellow points will give rise to circular expanding waves with speed vy, where vy is larger than vd because the yellow cells divide faster. And in this model, the position of the front at the next time step is given by the envelope of all these circular waves. Now, if we do every step, infinitesimal step one at a time, we can solve this analytically. And the analytical solution for such a sector is that the sector should be described by a logarithmic spiral. Here, phi is the opening angle of the sector, phi 0 is the initial angle, r0 is the colony radius at time 0, so here. And you see that the opening angle increases as the logarithm of the radius with these two parameters here that are the speed of expansion of the two cell types. Okay, so it's a very elegant solution, and we thought, okay, we can test this. We can measure the speed Vy by growing a colony made only of yellow cells. We can measure the speed Vb by growing a colony only of blue cells. And then we can test. So what we did is we went and measured all these opening angles for many of these sectors. And we plotted how the opening angles grow as a function of the radius. Now, if you look at the data, you can kind of tell that this is not a logarithm. The concavity is wrong. So we can plot the prediction according to the constant speed model, and it turns out to be very poor. And what I would like to convince you now is that it's very poor because it neglects the fact that this is a dense colony where cells are pushing each other. So we need something more. So what we did was we started thinking about these colonies as viscous fluids. The way we think about this is a colony is a viscous fluid made of two different phases, where the two phases correspond to the two colors, to the two strings. In this fluid, we assume that it's not only the cells at the edge that are actively dividing, but if you go and measure, we can measure where cells are dividing, and it turns out cells are dividing within a layer of about 500 microns. So the region of cell division is quite large compared to the diameter of the cell, which is only 5 microns. So we take this into account. This will be a source term for our fluid, because this is a self-reproducing fluid. It's an active fluid in a sense. In this fluid, cell division causes the local accumulation of pressure. So regions of the fluid where cells are dividing fast will correspond to regions of high pressure. And regions of the fluid where cells are dividing more slowly will correspond to regions of lower pressure. And in response to this pressure, this fluid flows.
So, Andrea, why is there no cell division in the center still? Because aren't the cells still active and able to divide?
So they are able to divide, but what happens is that if this is the agar, the colony starts out here. And so it depletes the nutrients in the middle very fast. And at steady state, this establishes a layer of cells that are actively dividing. It's a bit like a prevascular cancer that has a necrotic core. It's, in some sense, this is the necrotic core of this colony. Thanks. Yeah. Okay, so now this is a cartoon of the model. This is how we model these things in practice. If you look at the colony from the side, it looks a bit like this. It's a very, very short cylinder. Since it's a very short cylinder, we wanted to get a two-dimensional model. So to get the equations, we start from the continuity equation of this fluid. For the fluid dynamicists in the room, G here is not gravity, it's a growth rate function. So on the right here, we have the source term for this fluid, and the assumption we make is that this growth rate function is a function of space that's only different from 0 on the outer edge of the colony, within a distance delta from the perimeter. If we have two cell types, G might change value because yellow cells divide faster than blue ones. The density within the colony is relatively constant, so we assume this is an incompressible fluid. Now we are still in 3 dimensions. We can combine these two equations and get an equation for the divergence of the velocity field. And for those of you that are familiar with fluid dynamics, this is quite common. As for the momentum, we assume this is a viscous fluid, so we disregard inertia, and so we describe the dynamics using Stokes' flow. Up to now, this is still a 3-dimensional fluid, and here mu is the dynamic viscosity of this fluid. So the way in which we get a 2-dimensional model is by using what's called the lubrication approximation for thin films of fluid. And we also assume that the colony has a constant height, which is not true, but it's a relatively good approximation. This allows us to go from a 3-dimensional model to a 2-dimensional model where all quantities are averaged in the z direction. We get still the divergence of the 2-dimensional velocity field is equal to the growth function averaged in the z direction. And instead of having the Laplacian of the velocity being equal to the gradient of pressure, we recover what's known as Darcy's law. So we find that the velocity is proportional to the pressure gradient. And we are recovering Darcy's law in settings that are quite different from where we usually find it, which is in the flow equals. So we have a friction-like term here that depends on the dynamic viscosity of the colony and on the height of the colony. And these are all parameters that we can measure. So this is our model. This is our 2-dimensional model. From now on, I will drop the indexes to make things simpler, but from now on, we are in 2 dimensions. So if we take the divergence of the second equation, we get a Poisson equation for the pressure field. We find that the Laplacian of the 2-dimensional pressure is equal to minus the friction term times the growth rate function. And if we have a simple circular colony of just one strain, we can solve this analytically for the speed of expansion. We need a boundary condition for the pressure, which is constant pressure at the boundary. And we find that the expansion velocity of this colony, this fluid expanding due to pressure buildup, is given by the typical growth rate of the strain we're considering. This is related to the cell division time, times the growth layer width, times a factor that's negligible in our experiments, because typically the radius of the colony R is much larger than the growth layer width delta. So in practice, the expansion velocity is just the growth rate times the growth layer width, which if you think about dimensionality makes a lot of sense.
So is that pressure term related to surface tension or something that's keeping the front from moving?
Yes, that's a fantastic question. So we believe that surface tension here is not terribly important. We have some experiments to show that this is the case. Basically, we artificially create very large curvatures. We kill all the cells. Well, we don't really kill them. We stop their division, and we never observe this any kind of relaxation. So it's mostly pressure buildup due to the cell division within the colony.
What is it? Pressure, so it's outward pressure.
Yes.
Ah, okay.
Yes.
So it's a boundary condition.
Yes. Yeah. So in the sense that the boundary, they are exposed to atmospheric pressure. So there we assume that it's constant.
Okay, right.
So this equation is useful for us because we can measure the cell division time, and so the growth rate, we can measure the growth layer width, and we can measure the expansion speed, and it turns out that it matches quite well. In fact, this doesn't depend on the assumption about Stokes flow. This must be true for an incompressible fluid in 2 dimensions. Okay, so that's good. But we're interested in multi-strain colonies. So once we have a colony that has 2 colors, 2 strains, we resort to numerics to solve this. We now need to represent the interfaces, because we need to know that the growth function changes value. The way we do it is using level set methods, in case anyone is familiar with them. Then once we have the configuration and the interfaces, we need to solve for the pressure by solving the Poisson equation. We then need to compute the velocity by taking the gradient of the pressure, and we can now update our interfaces. And we repeat this process over and over numerically. So if we start with a simulated colony that looks like this, with 2 large yellow sectors here and 2 small yellow sectors here, we can compute the pressure, and you can see that the pressure depends on the size of the sector. Large sectors give rise to larger pressures than small sectors do. And this is going to be key, because if we then look how these sectors expand, it turns out that smaller sectors initially expand more slowly than larger sectors do. And we get sectors that look at least like the ones that we see in the experiments. If we compare this to the geometrical optics, the constant speed model, you see that sectors are much larger in the constant speed model. And this is because it neglects the fact that these small sectors here are not capable of producing such a high pressure gradient to expand as fast as larger sectors.
Could you find like say a boundary value? So if the sector is smaller than this value, then it's going to die out?
Yes, that's a very good question. In this situation, the thing is that in this deterministic model, if a sector belongs to the yellow strain, it will always expand. But conversely, I'm anticipating a bit, you'll see, if you look closely here, there is a thin blue filament. That filament is going to survive for very long times, it turns out, but eventually it will go extinct. So the situation referred to would be described by this one. Although at some point that filament is going to become very small. It will be comparable to the diameter of the cells. And at that point, I wouldn't trust this model anymore. But one can do agent-based models in which cells are dividing and pushing each other. And in there, you can recover these sectors and you can estimate how long it takes for them to go extinct.
So since you have 2 phases, your level set formulation uses 2 level set functions?
Exactly, yes, yes. Which
And do you often have to pre-initialize the whole thing because they start to cross over?
Yes, yes. There are a few tricks we need to do to avoid that. I even forgot at this point what kind of tricks I used, but that is an issue. But there are methods to avoid that from happening too often. Okay, so we can measure all the parameters of this model independently. We can measure the viscosity, we can measure the colony height, we can measure the growth rates, and it turns out that this model can really predict quite well the expansion dynamics of these sectors. So mechanical interactions are important in this context. The geometrical optics model fails to describe the dynamics because it neglects the fact that small sectors cannot produce a high enough pressure to propagate at the same speed as larger sectors do. There's another thing that I think is interesting, which is we saw numerically that if you send the growth layer width delta to 0 while keeping the expansion velocity constant, the continuum model tends to the geometrical optics model, which makes sense, right? If the growth layer width is 0, then we are in the domain of the geometrical optics model. And we can test this experimentally. We found a way to reduce the growth layer width by changing the agar concentration in the plate. The reason why this is true, like why 2% agar plates should have a smaller growth layer width than a 1% agar plate, it's, I would say, a bit unknown, but we can measure the growth layer width, and it turns out to be true. And if you do experiments with a smaller growth layer width, you'll see that the opening angles open up much faster. In fact, the constant speed model gives a good prediction for the rate at which the opening angle increases when the sector is large, so in the limit of large radii or large sectors. All right, there's another thing here that's interesting, which I've a bit alluded to, and it's what's going on with those sectors there. If you look in between the 2 yellow sectors, there is a very thin blue filament that survives for incredibly long times. If this picture here shows you the fluorescent channel corresponding to the blue cells. So blue cells look white here. If you look here, there are still blue cells at the very, very edge of the colony, even though they are dividing more slowly than the yellow ones. We would expect this to go extinct pretty fast. It turns out that they are benefiting from the pressure generated by the yellow cells. So this is a mechanical interaction that forms these long, we call them filaments, that survive for very long times. And the reason why they do is that if you have a very thin filament like this one here or this one here, it's so small that it doesn't change the pressure very much. It enjoys the high pressure generated by its neighbors. In a sense, it's being squished and pushed towards the exterior of the colony. And that's why it can propagate at the same speed as the yellow selectors. It will go extinct eventually, but it takes an incredibly long time. Each cell is 5 microns. These filaments can go on for centimeters. Okay, so this is the end of part 1. What I hope I convinced you is that mechanical interactions reduce the power of natural selection in 2 ways. On the one hand, they enhance the survival time of these low thickness strains through this mechanical effect by pushing these cells towards the edge. And on the other hand, they slow down the rate at which fitter strains take over the population in the initial phases of the dynamics because they are unable to create sufficient pressure to expand fast. This is a good time for questions, if anyone has any.
Growth rate, what does that look like through the growth layer? Is it slowing down as you get closer to the core? Or how do you model that?
Yeah, so in a sense, having a 2-dimensional model, we don't care too much about it. What people see is that it's the cells at the bottom that are actively dividing. And as the colony becomes thicker, it stops. For us, it's not terribly important because the edge of the colony is quite shallow. And eventually, in the interior of the colony, there is some residual growth, and cells will continue to pile up, but at an incredibly smaller rate. And that's where what happens to the bottom with respect to what happens to the top is important. In a sense, cells that are on the bottom have more access to nutrients and less access to oxygen. Cells that are on the top have more access to oxygen but less access to the nutrients. And this causes a gradient whereby it seems it's the cells on the bottom that are dividing faster than the ones on the top. Okay, so someone might call this ecological dynamics. I have shown you no mutations. It has implications for evolutionary dynamics because this sector might actually be a beneficial mutation that makes cells divide faster. But there is no mutation going on here. So right now what we're doing in the lab is we are looking at evolutionary dynamics through accumulation of mutations. This is work that is being done by Iad Amvi in my group. What we're doing is we're repeating the same experiment. So up to here, the experiment is the same. We start with a liquid culture, we inoculate on the agar plate, we grow it up as a colony, but now what we do is we select the cells at the edge, and we start a new colony with those cells. We let it expand, and then we do the process over and over again, a bit like the Lenski experiment, except that now it's in space. And what we find is that if cells initially looked like this is the very edge of the colony through light microscopy. Each individual circle here is an individual cell. This is how the ancestors look like. This is how yeast cells are supposed to look like. The evolved cells now look like this. This can happen even after 5 transfers, so it happens very fast. It happens in all our replicates, so we strongly suspect that cell shape has something to do with how fast the population expands. All the cells that we find now in the experiments are almost rod-like. They are also a bit larger than the ones that they are ancestral. And what we believe is going on is that these cells, if you look at them, they pack extremely closely. They pack very well. These cells, if you look carefully, you'll see there are many places where they're not packing as well. And if they leave more empty space behind, we believe they will expand at a faster rate in 2 dimensions. So we are now trying to understand this using agent-based simulation in which cells are actively pushing, dividing and pushing each other. and by having cells of different shapes, of course.
Is the speed of expansion the same at every radius of the colony? Could it be, because an alternative, which seems possible, is that you want to be of that shape when you are out there at the edge once the colony is certain radius?
Yes, so the speed of expansion is approximately constant as the colony expands. So if you measure the speed of expansion, it's higher initially, and then it stabilizes for a while, and then eventually drops down when the nutrients are decayed. We have not, so a way to test this would be to do the experiment and selecting cells very quickly. So we haven't tried to do that. But we could. I don't know. So you mean that this would allow them to fill up the space more quickly?
Perhaps.
When the curvature
Once
is
you are
smaller.
away from the center. So in the very beginning, when you're just starting the new colony, these elongated ones, is it expanding already faster than the first one did, the one which started with circular cells?
Yes, yes, very visibly so. Yeah.
Okay.
So there's a confounding variable though. These cells, I'm showing you, here it's very striking, they are evolving a different shape. They are also accumulating biomass more quickly. Like the Lansky cells, the growth rate is becoming larger. And experimentally, what we are doing right now is we can change the cell shape without changing the growth rate by introducing some mutations that affect cell shape without affecting growth rate too much. So we can test this, but we don't have the data yet.
Quick question. If they're not packed very well, does that mean their porosity is increasing so they're better able to oxygenate themselves and metabolize?
It's possible, yes, it's possible. Typically, these colonies are wet by a layer of water. So it's not that oxygen would go through air, let's say, but
This is different than the yeast, as you said, the yeast, it mattered, exposure to oxygen, exposure to nutrients.
well, the top surface is always exposed, yes. So one thing, actually, yeah, maybe the answer is yes, because one thing about these colonies is that they are also shallow. We don't know if this is related to the cell shape, but for sure that will increase the access to oxygen. And maybe the open space in between might also favor the diffusion of nutrients throughout the colony.
Yeah, so if I look at a circle, if I just cut it into like 10 pieces, going from the outside to the inside, it seems like you're taking the outside and then regrowing them. But if I were to take it at each level, going into the, into the middle, do you see that, if I take the 9th portion, which is closest to the outside,
Yeah.
is that growing faster than the 8th and the 7th and the 6th? Do you see that for each group?
So I can answer this question partially. What we do in this experiment is, in one treatment we pick cells from the frontier, and in another treatment we pick cells from the center. The cells that we pick from the center are much slower. They are now expanding much slower. They are also much more diverse. We don't exactly know why, but in the middle, in the population that we select from the middle, we see a lot more shape diversity. Some cells are round, some cells are slightly elongated. So the answer to the question, I guess, is if you select from the center, they grow more slowly. Okay. So this brings me to part 2. I've shown you one way in which spatial structure can emerge. We found that the expansion dynamics itself can cause different colors to separate. People realize that biological interactions can also affect how different colors separate, how different genotypes are distributed in space. For example, Melanie Mueller created 2 yeast strains that cross-feed essential amino acids to each other. Now, these cells cannot grow in isolation, and so if you grow a colony of them, first of all, it grows much more slowly, but also the colors separate much less than they do here because they can't. If they separate, they cannot grow anymore. On the other hand of the spectrum, people at Georgia Tech, McNally and others, they repeated similar experiments with strains of Vibrio cholera. Vibrio cholera would naturally stab its neighbors and inject toxins into them. If the neighbor is a relative of the same strain, nothing happens because they have the antitoxin. But if the neighbor is a different strain of Vibrio cholera, it dies. So they didn't focus much on what happens after color separation, and they were looking at what happens inside, in the interior of the homeland. And what they found is that if you switch off the antagonistic interaction, you get a relatively well-mixed population. But if you switch on the antagonistic interaction, you get coarser domains. You get the formation of these domains that are either all blue or all red. The idea here is that this is a bit like oil and water. There is a surface tension, an effective surface tension by which this cluster wants to minimize the perimeter to area ratio. And we thought that this was really interesting because it should mean that this system should also exhibit what's known as nucleation dynamics, which briefly this means that if you have a landscape that's completely colonized by a resident strain, which I'm showing here in cyan, and assume that this strain is constantly producing a toxin to fight external invasions. If we now introduce another strain that produces a toxin, and let's say it's stronger, it produces more toxin per unit time, we would expect it to replace the resident population. But this only happens if the amount of cells you introduced is above a critical nucleation threshold. So we wanted to test this experimentally, so we created a system working with yeast. We can make yeast export toxins into the environment. What you're seeing here is a toxin producer colony that we created next to a sensitive colony which doesn't produce the toxin and is sensitive to the toxin produced by this one. And this is grown on a petri dish that contains a stain that colors dead cells blue. So you see there's a lot of death going on at the interfaces, but the toxin can diffuse, and so we get kills also behind the petri line. I won't focus too much on how we do this, but I just want to communicate that what we did is we took a toxin encoding for a gene, we integrated into the genome of these cells in such a way, using an inducible promoter, that we can control the rate at which these cells produce toxins. This allows us to move in parameter space. We do the same for the other strain, where we use a different toxin and a different inducible promoter. We can vary how much K1 toxin is produced by varying galactose, and we can vary how much K2 toxin is produced by varying the concentration of copper. And we also integrated two different fluorescent proteins so that if we look at them at the microscope, one cell is magenta and one cell is cyan, so we can tell where different killer strains are. So how do we think about this system? If we think about a well-mixed population, like the experiments by Lenski, we can write down a simple equation for the abundance or density of cells of type 1. These are the K1 cells. We have a logistic growth term that has a carrying capacity that is shared by the two cell types. And then we need a death term, which is caused by the presence of the toxin produced by the type 2 cells, whose the density of that toxin, we are calling it C2. And of course, there's the corresponding equation for the abundance of type 2 cells. Then we need an equation for the concentration of the toxin. In that equation, we need a toxin production term and a toxin binding term. These are toxins that bind to the cell wall of cells. And we know for a fact that they bind to the cell wall of both cell types, but they only end up killing the type that is not producing. Here we could include a degradation term, but it turns out that they degrade very slowly, so we neglect that. And there's a corresponding equation for the toxin of type 2. Now, we don't like this model because it has a lot of parameters. We could fit this model to anything. But the good thing is that the concentrations of the toxins involved, hidden in these equations, there are two timescales. One for the concentrations of the toxins and one for the concentrations of the cells. it turns out that the time scale over which the concentration of the toxin changes is much, much faster than the time scale over which the population change. And so we can approximate this equation as being equal to 0. The underlying assumption here is that we are assuming the toxin concentration balances with the population density instantaneously. If we do this, we can simplify the model quite a lot, because we can get, instead of 4 equations, 2 of which I'm hiding, we get just 1 equation for the frequency f of the type 1 cells. This is the relative abundance. F is N1 divided by N1 plus N2. So this is a much simpler model. It only has 2 parameters, R1 and R2, which are combinations of the parameters of the full model. And these are parameters that we can measure. Now, if I write it like this, it becomes clear that this is a very simple dynamical system that has a cubic function of f in here. It has an unstable equilibrium here and 2 stable equilibria here. These 2 stable equilibria correspond to the extinction of one of the 2 states. The reason why we like our experiment is that we can move in the space of parameters. We can change R1 and R2. So if we make the magenta cells produce more toxins than the cyan ones, we are somewhere here, for example. And now the derivative looks like this. And you see that there's an unstable equilibrium here, which means if we start a well-mixed liquid competition with 40% of the magenta cells, the system will tend to the exclusion of the cyan cells. But even though the magenta cells are stronger, if we start to the left of the unstable equilibrium, the magenta cells will go to extinction. They are not strong enough to replace the resident population in these well-mixed conditions. And of course, if we move, then the equilibrium moves. We can also understand this as being the derivative of a potential function. The potential function will have a maximum at the unstable equilibrium, and the system might go left or right, depending on the initial condition. But we are mostly interested in spatial dynamics, and it turns out that this kind of equations have been studied for a very long time. We can use results by Rouhani and Barton if we look at this problem along an infinite line, where now we need to introduce a diffusion term that represents cells moving in space. It turns out that this system leads to bistable invasion waves if the initial condition is right. So if you have a sufficiently large inoculum, the stronger invading antagonist will replace the resident population by expanding as a traveling wave. This is a nonlinear wave. And it's a notable nonlinear wave because we can find a traveling wave solution. It might be, in case you're familiar with the Fisher-Kolmogorov equations and the Fisher waves, in there you can only find an exact solution for very specific values of the parameters. In this case, we have an analytical solution, and the analytical solution is of the traveling wave form. So the profile, the density profile, is a function of x at t. That's actually a function of x minus vt, where v is the speed of the invading wave. And the invasion wave speed depends on the parameters of the model, of course. So right now we are testing these ideas in the lab. One can go even further and build a stochastic partial differential equation of this scenario. If you account for the fact that birth and deaths are stochastic events, one finds that this equation should be complemented by a noise term, which is an interesting noise term because it's multiplicative. Here, eta is a Gaussian zero mean delta correlated noise. But its amplitude depends on the product of f times 1 minus f under the square root. It's a very interesting noise because it goes to 0 whenever f is either 1 or 0, which means that if we have a spatial configuration in which we have yellow cells and blue cells, this noise is really only active at the interface. So we can think of this noise as causing this cluster to change shape locally. And I don't have much time to go into the details of this, but one can understand this kind of evolution as a noisy evolution in the effective potential V that I showed you before. In fact, this is a system, it's the dynamics of the field that relaxes to a minimum of this V energy. And we can write down the free energy for a disk-shaped cluster. And the reason why this is interesting is that this free energy contains two terms, one that is proportional to the perimeter of the cluster and one that is proportional to the area of the cluster, which we can interpret as a line tension and a condensation energy. These two energies are fighting each other. And we can use this to find the critical size of a disk-shaped cluster, which turns out to be given by, again, a combination of all the parameters. So in the last very few minutes, I'll show you how we are testing this experiment. We have this system where we can control the rate of killing by the two cell types. We can parameterize our equation by doing competition assays between killer cells that are shown in colors and sensitive cells that are shown in gray. We can grow cells starting from 50-50 initial frequency and measure the relative abundance of the two cell types as a function of time. And we can use this information to fit our models and fit the values of interaction coefficients R1 and R2. As we increase the concentration of galactose, we have that the interaction coefficient R1 grows, and if we increase the concentration of copper, these are two different things, the concentration, the parameter R2 increases. So this is how we fit the parameters. We can then validate the model by doing competitions between two killer cells. And now we can ask, is there an unstable equilibrium? So we can do experiments where we start with a few cells of the stronger killer, and we can start competitions with more cells of the stronger killer. And if you do this and you plot the initial frequency of the stronger magenta killer on the x-axis, and the frequency of the same cell after 48 hours, this is what we see. This is the one-to-one line. The point where the line intersects the data is the unstable equilibrium. All points to the right, you see, increase their frequency as a function of time. Here we are to the right of the unstable equilibrium. All the points to the left decrease their frequency. We are to the left of the unstable equilibrium. And the curve here is the prediction of the model parameterized using the other independent experiments. So the model seems to work quite well. We do this at many different concentrations, and it seems to really work. However, we are interested in spatial dynamics. The spatial dynamics, for now, we've investigated like this. We took petri dishes filled with agar. We spread a loan of the cyan cells, which are the weaker killers, and then we introduce a droplet of the stronger killers in the middle. And we can do droplets of different sizes. Now, these are few of the sizes that we have done. You're looking at the petri dish from the top. On the left, you have the smaller inoculi. On the right, you have the larger inoculi. And we found a way to propagate this in time, because eventually what happens in this system is the nutrients go depleted and nothing happens anymore. But we can do a dilution that preserves the spatial structure. And as we do all our transfers, every column here is the same replicate evolving over time. And what's going on here is that indeed we do find a very sharp transition between inoculi that go extinct and inoculi that survive. There is a lot of dynamics going on here, and a lot of interesting things that I'm happy to talk about at any time. For example, if you look closely, there's a no cells length at the interface between the two cell types due to the diffusive nature of the toxin. The model I've shown you can reproduce some of the features, but not all. The model neglects toxin diffusion, and so it does not predict this, but we can understand this by making a more complicated model. So that's all I wanted to tell you. I think the main message for this talk is that we can use microbial microcosm to look at really cool math. We can use microbial microcosm to look at evolutionary dynamics in spatially extended landscapes, especially populations of cells that are dense. For these kind of populations, we need to borrow some tools from mechanics and some tools from soft matter physics. And in the case of the antagonistic interactions, we could borrow some tools from nucleation theory, which I think is kind of cool. As far as I know, this is the only example of nucleation theory in an experiment with microbes or with living organisms. So I'm quite excited about it. This is work that I did together with David Nelson and Andrew Murray at Harvard. Most of it, the work on the evolutionary dynamics, is being done by Iad Amvi in my group. And for the students, in case you like this talk, I'm teaching this class, which is a class on stochastic processes in biology, which I go through some of the things I've showed today. Thank you.
Okay, we have a few minutes for some questions, so if there
And the last experiment you just showed, seems like you guys drop it in the middle. If you drop it on the edge, do you get, like, does that affect whether it sort of colonizes?
It probably will, yes. So the way in which we do it is we do a few colonies per plate, but the distance between different colonies is very large. So if this is the inoculum, we separate them by at least 4, even 5 diameters of the inoculum, just to simulate having an infinite landscape of an infinite populations of the Sahian cells. I believe that if you were to do this at the edge, then you would have to worry, if this is the edge of the plate, then you would have to worry about the fact that these might have an easier time invading here, because there's going to be a smaller flux of toxin coming from that side. We haven't tried.
So in the experiments, all of the inoculi looked like they were roughly circular.
Yes.
Is it sensitive to shape if you took the same amount of the invading material and made it an elongated oval or something?
Yeah, it should be. We haven't tried it. We want to try at some point. We would like to try to do a linear inoculation. Surprisingly, it's much harder to do a linear inoculation than it is to do a circular one, because the circular one you can just like that. The reason why we would like to do a linear one is that then the surface tension should not matter. In that case, you have no curvature. But we haven't tried. But it should be. It should be.
So I'm a little confused. Your experiments in the last one seem to show that those which do not go extinct, they stabilize in size.
Yes.
Yet the traveling wave solution does not predict that. Is this just the effect of the finite size of your experimental dish?
No, it's so these are the areas of those inoculations as a function of time for all replicates. What happens is that initially both cell types retreat. They form this no cells land, and eventually they start growing, and some of them grow and start expanding further than they started. The reason why the dynamics is so 12 transfers here means 12 times 10 days. So this is an experiment that lasts for 120 days. Now, what's going on is that cells do not move much. So the diffusion coefficient of the bistable invasion wave is extremely slow. And so the dynamics of growth here is so small that it's hard to tell from the picture. But they do grow eventually. We have some other interesting thing. There is an outlier here. Above the critical size, it went down. And this is associated with a mutation that occurred that surprisingly made the invader cells produce fewer toxins. We don't completely understand how this is possible. We suspect that in combination with that mutation, the resident strain became resistant as well by looking at the colony. Although when we selected from the edge, we couldn't find resistant mutations. We believe that we might have missed it when using a toothpick to select cells. But they do expand. So the problem with this system is that the way we do the spatial dilution is we take a cylinder, we put the microfiber cloth on top, then we take the cell, the paper dish, we press it onto there, then we take a fresh petri dish and we press it again, and that is our dilution. It's a bit noisy in the sense that cells can get squished a little bit. So to study the bistable wave dynamics, what we're doing now is we are growing cells on set individual beams in devices that are called 96 well plates. And we pipette cells left and right to simulate the movement dynamics. So in that way, we can impose the diffusion coefficient. And the idea is that we will use that to look at the pushed wave invasion dynamics in this system in a more controlled way. This, unfortunately, is semi-quantitative. Like, if we can model this by having toxin diffusion, cell diffusion, and measuring all of these parameters, and we understand that this should be the critical radius, right, like we see here. But it's just hard to do more than that.
Okay, well, that was a wonderful talk, and I hope that I invite everybody to thank our speaker in the last